this session, we will learn some aspects about pedigree drawing and patterns of inheritance. The learning objectives of this session include some basic tenets about pedigree as a tool that is used in medical genetics, and some overview about the various inheritance patterns, including Mendelian patterns and non-Mendelian patterns of inheritance. So pedigree is an important tool that is used in clinical genetics, and the word pedigree has come from the word Pedigree, which is a French word which means crane's foot. So pedigree is a graphic representation of a family's health history and genetic relationship. So initially, when pedigree was used as a tool, instead of straight lines, curved lines were used, and this gave the resemblance of the foot of a crane. So that is how the word pedigree has been used since then. Some of the basic aspects of pedigree drawing include certain lines that are described, and the information that should be contained in the pedigree should be from 3 generations. And these 3 generations are usually named in Roman numerals, and the individuals are named in Arabic numerals. And a square is a symbol that is used to depict a male, and a circle is a symbol that is used to depict a female. Affected individuals are usually depicted by shading or coloring. And if there are multiple conditions which are seen in an individual, they can be depicted by using different types of shading or different types of lines. And the relationship line is a line that connects 2 individuals, as you can see here in this part. There is another line which is known as the line of descent, which will connect one generation to the other. And there is a line known as the ship line, which will connect different siblings. The person who actually has the phenotypic feature, or who comes to medical attention for the first time from a family, is known as the proband, and it is depicted, and he or she is depicted by the letter P. Consultant, who is depicted by the letter C, is the person who comes for genetic counseling, or who the consultant may or may not be affected. So in case of affected children, usually the parents will be the consultant. So there is another line known as this individual line, which will connect each particular individual to the pedigree. These are some of the other symbols that are used in pedigree drawing, and an extensive review about the standard terminologies and the symbols that are needed to be used is provided in this paper, which was published in Journal of Genetic Counseling in 2008. Now, coming to genetic diseases can be broadly divided into 3 categories. One is chromosomal, the other one is single gene disorders, and the third one is multifactorial disorders. So in the subsequent slides, or in this presentation, we would be mainly talking about the patterns of inheritance of single gene disorders. So single gene disorders implicate or imply or refer to conditions where the genetic factors provide or act as a major contributing factor. And we all know that it was Gregor Mendel whose work enabled us to understand genetics in its current form. And to honor him, the patterns of inheritance are named as Mendelian patterns of inheritance, and single gene disorders are also known as Mendelian disorders. So the patterns of inheritance may be broadly classified as Mendelian and non-Mendelian. So by Mendelian, what we mean is the group of inheritance patterns which actually follows the laws of inheritance proposed by Mendel. And these include autosomal dominant, autosomal recessive, X-linked and Y-linked inheritance patterns. There are other patterns of inheritance which do not follow Mendel's law, and these include mitochondrial inheritance, unstable repeat expansion, genomic imprinting, uniparental disomy, mosaicism, and multigenic inheritance. Some of the definitions that we need to know before we proceed include what is a locus. So locus is actually the position of a gene on a chromosome. Here you can see that there are two loci, locus 1 and locus 2. Locus 1 has gene A and locus 2 has gene B. And allele is an alternate form of a gene at a particular locus. So here in locus 1, there are two alleles for gene A. One is capital A and the alternate form is small letter A. Similarly for the locus B. Now, autosomes include all the chromosomes other than the sex chromosome, and an individual is considered to be homozygote if he or she has the same allele at a particular locus. And if two different alleles are present at a particular locus, then he or she is known as a heterozygote. Now, coming to the terms dominant and recessive, this is with pertaining to the phenotypic features. So, if the phenotypic feature expresses only when an individual is a homozygote at a particular loci, then that phenotype is known as the recessive phenotype. And if an individual expresses the phenotype both in heterozygous state as well as in homozygous state, then that phenotype is known as a dominant phenotype. So coming to the first pattern of inheritance, which is autosomal dominant inheritance. So here the variant or the mutant will be on an autosome. So that is why the term autosomal. And dominant means that in both heterozygous as well as homozygous states, the phenotypic features are manifested. And when the phenotypic features are same for a heterozygote and a homozygote, we classify such conditions as pure dominant conditions. And when a homozygous individual has a severe phenotype compared to a heterozygous individual, we classify it as incompletely dominant conditions. Some of the common autosomal dominant disorders, examples include achondroplasia, Marfan syndrome, tuberous sclerosis, etc. So this slide shows the various features of an autosomal dominant disorders in a pedigree. So here you can see that multiple generations are individuals and multiple generations are affected, and this is known as vertical transmission. And both males and females will be affected in equal proportion. Male to male transmission is usually seen. There are certain features which an autosomal dominant disorder can exhibit. One of them is known as pleiotropy, where a single variation in a particular gene gives rise to effects in different organ systems. For example, variation in tuberous sclerosis 1 or tuberous sclerosis 2 gene actually can result in phenotypic features in the brain, in the skin, kidney, or lung. Another feature that can be demonstrated in case of autosomal dominant conditions is variable expressivity. Here, individuals having the same genotype may express different severity of the condition. For example, in this pedigree, a family having a variation causing autosomal dominant polycystic kidney disease can have individuals with different severity of clinical conditions. Some of the autosomal dominant conditions exhibit a phenomenon known as decreased penetrance or incomplete penetrance. So penetrance is the proportion of individuals who express a particular phenotype and carry the same genotype. So here in this pedigree, the individual who is marked here, who is 2-4, has the variation but does not manifest the phenotypic features. So here there is non-penetrance of this particular phenotype. Now coming to the recurrence risk in autosomal dominant conditions, an affected parent or an affected individual, if he has an offspring with an unaffected individual, the chance that the variant or the mutant allele is transmitted to the offspring is 50%. The next pattern of inheritance that we would discuss is autosomal recessive inheritance. So here, an individual who is homozygous or compound heterozygous for a particular variation will be affected, and the heterozygous individual who carries only one allele, one variant allele, is usually asymptomatic and is known as a carrier. Some of the features of autosomal recessive inheritance in a pedigree include multiple individuals who in a particular generation who are affected, and this is in contrast with multiple individuals in multiple generations who are affected in case of autosomal dominant inheritance. So here, this pattern of inheritance is otherwise known as horizontal transmission because multiple individuals in a single generation may be affected. Consanguinity may be seen, and both males and females are affected in equal proportions. Consanguinity is the union between two individuals who are as close or closer than second cousins, and they inherit an allele from a common ancestor. And the chance that both parents are carriers for a particular disease increases. Consanguinity is more likely to precipitate very rare genetic diseases. The recurrence risk in case of an autosomal recessive condition when two partners are carriers is 25% chance of their offspring having being affected with that autosomal recessive condition. And if there is an affected parent who already has a biallelic variation causing a recessive disease, if that parent has an offspring with another individual who is the carrier for this same condition, 50% of their offsprings will be affected and 50% of their offsprings will be carriers. This phenomenon is known as pseudo-dominance. Some of the common examples for autosomal recessive conditions include the spinal muscular atrophy, beta thalassemia, etc. Now, coming to X-linked inheritance. So here, whether an X-linked condition is dominant or recessive depends upon whether the phenotype is expressed in a heterozygous female, and the phenotypic expression in a heterozygous female also depends on pattern of random X inactivation and the proportion of cells in the pertinent tissues that have the variant allele. Now, some scientists are of the opinion that X-linked recessive or X-linked dominant terms should not be used anymore. They should be considered as a spectrum of disorders. When there is no penetrance in a carrier female, those disorders fall on the extreme left side and can be classified as X-linked recessive disease. And on the extreme right, you have X-linked dominant conditions where there is a full penetrance in a heterozygous female, and there are other conditions in between with variable penetrance. Some of the features in an X-linked pedigree would include, so here this pedigree depicts an X-linked recessive pedigree. So here the affected individuals are all males and carrier females carry the variant, but they do not have any phenotypic manifestation. And here male to male transmission is not seen, and a male transmits the disease to another male through the intermediary of a non-affected carrier female. So in X-linked recessive conditions, affected females may be there in certain conditions, like when the female has Turner syndrome and has only one X chromosome, when there is skewed X inactivation, when there is X autosome translocation, and when there are biallelic variations in a gene on X chromosome, or in case of a phenotypic female as in complete androgen insensitivity syndrome. So in case of an X-linked recessive disease, if there is an affected male and an unaffected female, then all the daughters of that unaffected, all their daughters will be carriers, and none of the sons will be affected. In case an unaffected male has an offspring with a carrier female, then 50% of their sons will be affected and 50% of their daughters will be carriers. Some of the common examples for X-linked recessive disorders include Duchenne muscular dystrophy, hemophilia A and B. Now coming to X-linked dominant inheritance. So here, like autosomal dominant pedigree, multiple individuals in multiple generations can be affected, and males and females can be affected, and male to male transmission is actually not seen. So that is the point that distinguishes this particular pedigree from an autosomal dominant pedigree. Some of the examples for X-linked dominant inheritance include Rett syndrome, incontinentia pigmenti, etc. Now coming to Y-linked inheritance. So here the traits, the phenotypic traits would be passed only from a father to his son. And some of the Y-linked traits include hairy pinna and some of the genes which are related to fertility. So now we will discuss about mitochondrial inheritance, which is non-Mendelian form of inheritance. So some of the features of mitochondrial inheritance include maternal inheritance. So this means that the mitochondria are derived exclusively from the ova or from the mother. Then there are other features like replicative segregation, homoplasmy or heteroplasmy, threshold expression, and bottleneck hypothesis, which we will discuss in subsequent slides. So mitochondrial inheritance follows maternal inheritance. So in a pedigree, here you can see that in case of an affected mother or a mother with a mutant mitochondria, all her offspring actually have mutant mitochondria, will inherit the mutant mitochondria, but an affected male with a mutant mitochondria does not pass the mutant mitochondria to his offspring. So that is why it is known as maternal inheritance. So in a cell, actually, there will be around thousands of mitochondria, which will mitochondrial DNA molecules, which will be distributed in hundreds of mitochondria that are seen in the cell. So some of the mitochondria may have only mutant DNA, or some of the mitochondria may have only wild type DNA. And when a cell divides, the daughter cells also get different number of mutant and wild type of mitochondria. And the mitochondria will be assigned to each of the daughter cells randomly. And this process is known as replicative segregation. And only when the number of mutant mitochondria is beyond a particular threshold, phenotypic expression would be manifested. So this phenomenon is known as threshold expression. And when a cell, when a daughter cell has only one type of mitochondria, that is either the mutant type or the wild type, that phenomenon is known as homoplasmy. And if there is a mixture of both wild type as well as mutant mitochondria, that phenomenon is known as heteroplasmy. Then another phenomenon that is discussed in mitochondrial inheritance is genetic bottleneck. So here, before the oocytes are formed, there is actually a decrease in the number of mitochondrial DNA. And after the oocytes are formed, the mitochondrial DNA again will replicate. So which of the ova actually contain variant or mutant mitochondrial DNA is a random process. So the number of mutant mitochondria that is actually inherited by each offspring will vary. Now the next pattern, which does not follow Mendelian principles of inheritance, include unstable repeat expansion. So these disorders have an unstable expansion of repeating units of 3 or more nucleotides in tandem. And the wild type alleles are usually polymorphic with variable number of repeats in the general population. These disorders can demonstrate a phenomenon known as anticipation. As you can see in this pedigree, the age of onset of disease is earlier as generation progresses and as the number of repeats increase. Some of the examples for these group of disorders include triplet repeat disorders like Huntington disease, Fragile X syndrome, myotonic dystrophy, and Friedreich's ataxia. Genomic imprinting is another non-Mendelian form of inheritance. So generally, phenotypic expression of an allele does not depend upon the parent of origin, but there is a situation known as genomic imprinting where the phenotypic expression of the allele depends upon the parent of origin, and it is an epigenetic phenomenon. And in the human genome, there are around 80 genes in differentially methylated regions. Some of the examples for genomic imprinting related disorders include Angelman syndrome and Prader-Willi syndrome. There is yet another phenomenon known as uniparental disomy, where sometimes both homologous chromosomes will be inherited from the same parent. And generally, uniparental disomy does not lead to any clinical significance, does not have any major clinical significance, except when that region has imprinted genes, or when there is due to homozygosity or due to uniparental disomy, it results in homozygosity for an autosomal recessive allele. And whenever there is an error in meiosis 2, it can result in uniparental isodisomy. And whenever there is a defect in meiosis 1, it will result in uniparental heterodisomy. Mosaicism is the presence of more than one cell type, which are derived from the single zygote. And this occurs because of an error in mitosis after conception. Mosaicism can be somatic or it can be germline. So germline means mosaicism happening in the germline cells in the testis or the ovary. And mosaicism may result in variable severity of clinical features. Now, coming to multigenic inheritance, which is the last one among the non-Mendelian pattern of inheritance. So here, the phenotypic effect occurs due to the combined effect of more than one gene. And the simplest of this multigenic inheritance is a digenic inheritance, where the phenotypic effects of two genes will combine together to result in a disease or in a phenotypic manifestation. And an example for digenic inheritance would be retinitis pigmentosa. Then you have, there are conditions which are otherwise known as like complex traits, where multiple genes may have an additive effect on the phenotype, and there may be environmental factors also which result in the phenotypic manifestation.